Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 24th November 2020. As all of us know that we are revising chapter number 12, biotechnology and its applications. In that we were talking about various applications in different fields including medicine, and in our previous session, we talked about its implications in the field of medicine, especially using gene therapy and molecular diagnosis. Under molecular diagnosis, if you recollect from your memory, we were talking about recombinant DNA, we were talking about PCR, polymerase chain reaction, we were also talking about ELISA, that is, I also said that this is normally for one more question, expansion, enzyme linked immunosorbent assay, that is ELISA. So, having discussed all this, now we move ahead with transgenic animals. We talked about transgenic plants, and now it's the time to talk about transgenic animals. What are these? Animals that have their DNA manipulated, manipulation of DNA to possess and express a foreign gene are known as transgenic animals. Transgenic mice, rats, rabbits, pigs, sheep, cows and fish have been produced, and the common reasons for development of transgenic animals are normal physiology and development. They are designed to allow the study of gene regulation, their effect on normal functioning of the body. By introducing genes from other species that alter the formation of this factor and studying biological effects that results. So basically, transgenic animals contain in its genome a gene, a single gene or group of genes introduced by one or the other technique of transfection. The gene introduced by transfection is known as transgene. In animals, transfection species or specifies the introduction of a DNA segment, that is gene of interest, either naked or integrated into a vector into an animal cell. This phenomena is known as transformation in all other organisms, but here we call it as transfection. So transgenic animals are produced because of the following reasons, and one of the reasons we talked about is normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functioning of the body and its development. Example, study of complex factors involved in growth such as insulin-like growth factor. By introducing genes from other species that alter the formation of this factor and studying the biological effects that result, information is obtained about the biological role of the factor in the body. So basically we are testing the physiology of an animal and introduction of new genes or gene of interest, how it affects the physiology and overall development of that particular animal. This is the main reason we are doing this. Second, study of disease. A number of transgenic animals are designed to increase our understanding of how genes contribute to the development of a disease. Transgenic model has been developed for diseases like cancer, cystic fibrosis, Alzheimer's disease, etc. So many transgenic animals are designed to increase understanding of a gene contribution to the development of a disease, how diseases are caused and how they spread, what is the gene that is responsible for this. These are specially made to serve as models for human diseases so that investigation of new treatments for diseases is made possible. That means we are making these animals, transgenic animals, as models for probable study of such a disease in human beings. That is the purpose of it. So today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, particularly because many people are suffering with this rheumatoid arthritis, and of course, Alzheimer's is becoming unfortunately very common nowadays. And then third factor is biological products. Transgenic animals that produce useful biological products can be created by the introduction of the portion of DNA, that is gene, which codes for particular product such as human protein like alpha-1 antitrypsin used for treating emphysema. Emphysema, recollect from your memory, lung disease, accumulation of water in the lungs and difficulty in breathing, chest pain, bulging of the lungs, etc. The first transgenic cow, Roji, this is the first one, Roji, produced human protein enriched milk. See, protein enriched milk. What is that? It contains alpha lactalbumin. How much? In 2.4 milligrams per liter. And this is a model again. Medicines required to treat certain human diseases may contain biological products, but such products are often expensive to make. Transgenic animals which produce useful biological products can be created by the introduction of the portion of DNA or genes that codes for particular product like human protein alpha-1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria, again another disease, phenylketonuria, PKU, and cystic fibrosis, very dreaded disease, cystic fibrosis. In 1997, the first transgenic cow called Rosie produced human protein enriched milk. We mentioned 2.4 grams per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. That's why we are able to use this technology to produce biological products. And most important, which we always talk nowadays, vaccine. In the COVID-19 situation, everybody is anxiously waiting for the vaccine for corona. So how effective, how safe is the vaccine that is going to come? So vaccine safety, transgenic mice are being developed for use in testing the safety of vaccines before they are used on humans. You must have noticed that a lot of reports are coming from two companies. One is Moderna from USA and Pfizer. These companies, German as well as American companies, they have successfully conducted the trials, trial 1, trial 2, trial 3, and now they are injecting these vaccines into the mice and seeing the results. And that is why the companies like Pfizer and Moderna and of course the Bharat Biotech from Hyderabad, they are all doing all this for corona vaccine. And this is being done because before they are used on us, they need to perform the tests on mice. So these are being used to test the safety of the corona vaccine, as well as previously we did polio vaccine. So if successful and found to be reliable, they could replace the use of monkeys to test the safety of batches of vaccine. So it is for vaccine safety also transgenic animals are produced. And next one, chemical safety testing. This is known as toxicity or safety testing. Chemical safety is toxicity testing. The procedure is the same as that used for testing toxicity of drugs. Transgenic animals are made which carry genes that make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances and the effects are studied in detail. So, for toxicity testing also, transgenic animals are being produced. And now, what are the examples of transgenic animals? So, transgenic cattle, the only successful transfection technique in cattle is micro-injection of fertilized ova, which may either be recovered surgically or may be obtained from ovaries extracted from slaughtered cows and cultured in vitro. So, there is ethical issue involved in that, okay, because we are slaughtering the cows and cultured in vitro. The chief objectives of transgenic cattle production are increased milk and meat production, molecular farming, improved properties and proportions of casein in milk. Casein is a milk protein, all of you know. Regulate content of lactose and butter, fat in milk, and enhanced resistance to viral and bacterial pathogens. That is the reason why we are creating transgenic cattle. And second one that we think of is transgenic mice. Mouse is the most preferred mammal for studies on gene transfer due to its many favorable features like short estrus cycle and gestation period, relatively short generation time, production of several offspring per pregnancy, that is litter, convenient in vitro fertilization, successful culture of embryos in vitro at least for a period of time, production and maintenance of embryonic stem cells, availability of a diverse area of genetic stocks. These are all the advantages why we prefer mouse for creating transgenic mice. As a result, the techniques of gene transfer, transgenic production have been developed using mice as models. But then again, in all these ethical issues are involved. And then another animal that I can recollect is transgenic rabbits. Rabbits are quite promising for gene farming or molecular farming, which aims at the production of recoverable quantities of pharmaceutically or biologically important proteins encoded by transgenes. The human genes encoding valuable proteins have been separately transferred into rabbits, like the human genes interleukin, growth hormone GH, tissue plasminogen activator, alpha 1 antitrypsin. All these are being injected into the rabbits, and the rabbits are able to help us because we can increase the numbers transgenic rabbits. And some of the interesting facts that I would like to add here is the creation of a superbug and supermouse. First transgenic animal was mouse, which contained a gene for growth hormone GH. Growth hormone is GH. Okay, and this enlarged mouse was known as super mouse. And another one is Pseudomonas pituita, has been genetically engineered by introducing different strains of plasmids. It resulted in new genetically engineered bacterium that cleans the oil spills in the seas and oceans. Because of late that has become a big headache for us, how to clean the oceans and seas, in fact rivers, from the oil spills that happen due to this boating and ships and extraction of oil and gas. So this has become a big headache for us, how to avoid the water pollution. And thanks to this Pseudomonas pituita, because this is able to clean the oil spills, and hence it is called since it eats the oil, it's called oil eating bug, or since it has reduced our headache in clearing of all the oil foam on the water surface, it's called super bug. So thanks to biotechnology for creating super mouse and super bug, and thereby solving some of the environmental problems also. We may need to discuss whether we have rights to use such animals for our experimental purposes. Do have rights to change their genomes. And we are all, at the end of the day, if you analyze, we are doing all this for our own sake. Of course, some of them are environmental issues also, but most of the time we are using for our own purposes. So does the ethics allow us? The question is that. We will discuss about these ethical issues and biopatency, biopiracy in our next session. Until then, keep on studying, increasing your knowledge, as knowledge is